Okay, Shweta madam, we can start.
Okay. So, good afternoon, all. It's my honor to welcome you all to this afternoon session on the third day of this AICT sponsored 2 week online faculty development program on innovation, startup, and entrepreneurship development in pharmaceutical sciences, organized by Kokan Ganpit Rahul Darkar College of Pharmacy and Research Institute, Karjat. Today we have with us Dr. Yogeshwar Bachcha. Dr. Yogeshwar Bachcha, after finishing PhD in pharmaceutics from Institute of Chemical Technology, Mumbai, worked in Europe for around 15 years with a different startup and mid-sized biotech and pharmaceutical companies. During his tenure, he has successfully managed pharmaceutical development of several projects from POC to phase 1, phase 2, and phase 3 clinical trials. He has a strong network of the experts and CROs, CMOs in Europe, in USA, and Australia. In addition, he has a 16 international publications, 8 patents, and 30 conferences proceeding to his credit. And recently, he has edited a book for Ville, VCH, Germany, on innovation dosage form design and development at early stage. Currently, he is heading pharmaceutical development at German biotech Alcuris Anti-Infective Cures GmbH, Wuppertal, Germany. As a consultant, he is also founder of pharmaceutical R&D consultancy firm Adex Pharma, which was established in 2006. The prime object of Adex is to resolve the complex issue in pharmaceutical development of new drugs and the life cycle management of approved drugs. Dr. Bachcha has invited as a speaker many international symposiums around the globe, like India, Europe, and the US. He is also a member of executive committee of the Controlled Release Society India chapter. And the topic is COVID-19 pandemic, basic treatment options, and CMC challenges. With this short introduction, I would like to Dr. Yogeshwar Bachcha to deliver the question. Over to you, sir.
Thank you. Thank you, Ms. Shweta, for the very kind introduction. But I would like to clarify the title of today's talk is Oral Delivery of Biologics. Maybe there's some confusion.
Yes.
So hope it goes well. I hope you all can see my screen. So first of all, very good afternoon to all of you. And it's my pleasure to be here to deliver a talk on this topic, oral delivery of biologics. So when we say about biologics, it really covers the vast majority of the compounds ranging from, let's say, peptides, proteins, antibodies. And so basically, maybe you can imagine in terms of the molecular weight, anything which is beyond, let's say, 1000, you can really try to put into the category of biologics. And then we do have limitations for the delivery of biologics. You cannot use the similar ways to transport these biologics, across the oral route as you might deliver the small molecules, which are having the molecular weight maybe in the range of 500, 800. You do have some very small molecules as well below, let's say, less than 500. And this is something to be considered. So in my today's talk, what I'm going to cover, so regarding the market scenario for the biologics, what are the limitations and challenges we face to deliver the biologics? What are the formulation considerations of formulation scientists is supposed to consider? Then what are the different options and what is work really going around, the glimpse from the clinical trials, and then we try to conclude. So when we think about market, so the market scenario is quite good, and there is a huge potential for the growth in this particular segment. Here I just tried to put together some facts. So from the year 2018 to 2026, you do have the annual growth rate of 33% more or less, which is quite huge. And then considering the market scenario, you have at least more than 300 monoclonal antibodies, 250 vaccines, and hundreds of other biologics, including the gene therapies or cell therapies, they are under clinical development. And what really drives this market scenario? So we do have increasing partnership and collaborations. To give very recent examples, I mean, the the COVID buzz is almost from last year we are hearing. More or less, it landed in India in March 2020, and we are still fighting against it. So a lot of companies got into this vaccine development, starting from BioNTech, Pfizer, Moderna. In India, we have Serum, then you have Bharat Biotech, etc. So you see the scope, considering the biologics. I don't have to really add. So I think more or less everyone is familiar. So a lot of innovative dosage forms and technologies are needed, because you can't just use a simple pill or capsule or solution to deliver this biologic. So you need a certain delivery there, and there's some innovation is needed. Most important, patient compliance and cost. So this is also something I can correlate to COVID scenario, that basically when you have to get a vaccine now, so we know then we don't get the slots, the vaccine is short. Why? The basic reason is they are given by the parental route. It must be sterile, it must be stored at, let's say, 2 to 8 degrees Celsius, and that has impact on the cost. Patient compliance, if I say, okay, then injection has to be given. If you have to get an injection, you must go to the doctor, right? And this is something then leads to the patient compliance issue. If it is, imagine if it is a tablet, as you have tablet for favipiravir or maybe some other, then it's quite easy. You just go to the chemist, you get that tablet, get it to home, and it's easy to administer. That's something, patient compliance. And all these scenarios are influencing in a way the biologics market. As example, I took his Soma, which is sort of delivery device for insulin. So self-orienting millimeter scale applicator. So what they are doing basically, so they have designed this device for oral delivery of insulin. So we know in importance of India, importance of insulin in India, because we are more or less the diabetic capital of the world. Maximum number of diabetic patients in the world we have in India. So a lot of patients, they need insulin on day-to-day basis. Type 1 it's must. Type 2 also, some of the patients, if they are not responding to other medication, then at the end of the day, then they need a daily insulin injection. And okay, if it is injection, again we are into the same issue as I said before, the patient compliance. So then we need some oral delivery system which could, make patients life easy. So this Soma was developed by MIT in Bob Langer's lab, and then they have collaboration with Novo Nordisk, Swedish company. The preclinical studies are going on, and they expect it to be in 2022. So this could be one of the example of the first bullet point, increasing partnership and collaborations. So until now, was it clear? I mean, are you guys able to hear me, see me? Just give me brief feedback, so that otherwise I'm just talking to myself, so that doesn't really help me out. Can someone just say so far so good?
You're perfectly
Hello?
audible with your screen. Your screen is also visible and you are also audible.
Okay, good. Thank you. Thank you for this quick feedback. Okay, then on this slide, I tried to list the blockbuster drugs, which are really driving this multi-billion industry or multi-billion market for the biologics. So it all started with this monoclonal. More or less, all these are, maximum. It's really comes from the monoclonal antibodies. Your big giants like maybe Roche, Pfizer, you have Genentech, etc. In India, we do have some companies that are doing biologics, but they are more biosimilars. In India, we have like Biocon, but they are most in the biosimilars. So the majority of innovative biologics, they are coming mainly from US and, let's say, some of the European companies. And you see here how the growth is really happening in the US dollars in billions, starting from 5.3 up to almost 20 billion US dollar, which is quite huge. And on this slide, I tried to put together, blockbuster biologics, which really help the pharma companies to get the significant revenues. But of course, we should also mind that developing a biologics is a huge, you know, task compared to development of the small molecules. So, I mean, I'll not go into the details, but to give a glimpse, when you have small molecules, you need to prepare a tablet or formulation. You just go with the simple synthesis, which maybe, 3 steps or 5, as maximum 20 steps. You have your molecule, you go for formulation. But imagine a biologics, then it's a totally different scenario. You have your downstream processing, where you start with the cell culture, and from the cell culture, the particular protein is expressed. When that protein is expressed, you need to go for a upstream process, and then at the end of the day, you get your protein molecule or the peptide molecule or maybe antibody, which has to be then formulated. And these molecules are quite fragile, they are not very stable, there are significant efforts are also needed to stabilize these biologics, and that's the reason the development of biologics is quite critical. And if you see the regulatory guidelines carefully, they are quite different for biologics compared to the small molecules. So just take a note of this slide. I will not go into the details. So which are the blockbuster biologics, in which segment they are using. Important aspect I can tell you, most of these drugs are used in the chronic situation where the small molecules may not be working, and that's where this biologics can bring some ray of hope, and they work out quite well because they work at a genetic level, right? So the mode of action is quite different for the biologics compared to the small molecules. And then we should also try to take into account the expirations of these biologics, because then that gives a scope to the biosimilar companies. So once these biologics, the innovated drugs or innovative biologics are out of patent, then the biosimilar companies can really get into the picture. They can try to determine the sequence, the protein sequence of these molecules. They can try to get into really the process. I told you before, the DSP and USP aspects, how you can try to make a proteins, and that can help them to come with the biosimilars, which are more or less the generic versions, you can say, of the innovator biologics. And this can be launched, and there is a possibility, let's say, to reduce the cost of the biologics. So let's get back to our main topic. So what is our aim? We would like to see whether this biologics can be delivered by the oral route, because you see majority of the biological molecules or macromolecules onto the market, they're given by the parenteral route, maybe by IV infusion, subcutaneous, intramuscular, right? And this is something maybe as a formulator we can think, okay, is it possible to give the drug by oral route? If yes, then what are our challenges? And this we can see here. Then when you imagine oral route, when any tablet capsule solution is taken by oral route, first it goes to stomach. What is the pH of the stomach? It's acidic environment, between 1.2 to 3.5, depending on the disease conditions, age. And then due to this acidic pH, as well as you do have some enzymes, these enzymes can lead to the degradation of the protein or peptide, because these are quite sensitive to the acidic pH. And that's where maybe then your protein is completely degraded, it is not available for the absorption, and then you have the impact. So imagine you are using very expensive drug, it is given by oral route, but if it is completely degraded, of course it's of no use, right? Then that's the first hurdle we saw. Then we do have the layer of intestinal fluid, and this also retards the passage of these molecules. Then you have the very tight cell membranes, which also restrict the passage of the biologics across the cell, the lining, I will say. So after we saw the barriers, in the intestine, across the GI tract, let's say the stomachs, and let's say what are our formulation consideration, what are the challenges. So as a free formulation study, we should try to take into account certain aspects. So the structural aspects of the protein molecule are very important. Then these are like primary, you do have the primary, secondary structures of the protein, which is quite critical for each protein. As soon as there's a change in the structure of the protein component, of course it leads to change of its the conformation. And if it's changing its conformation, then of course then it will not be able to bind to the receptor, because then the structure is very unique, and for the drug receptor interaction, that structure has to be intact. But if it is damaged, then the drug receptor interaction is not happening, and you will not be able to elicit that physiological response which is needed. I'm sorry, I will just say, hello, me jara call मध्ये नंतर phone करतो. I'm sorry for that. I'll just put on the airplane mode so that nobody is disturbing me. My apologies. So the second factor we should take into account physical parameters. So what are physical parameters? So these are mainly impacting the formulation part, because the solubility of protein is very important. Its hydrophobicity can lead to molecular aggregation or distability. Molecular weight is also important, because to deliver a molecule with 1500 would be easier, but imagine you have more protein molecule which has a molecular weight of 20,000 or 25,000. Then it's a different scenario to formulate to deliver. Self association is one aspect we should take into consideration. Aggregation state, glycolization. So basically these are all parameters which lead to the degradation or distability of the protein molecule to be considered. Then we go to the biological parameters. So how is the affinity of the protein towards receptor? When I say substrate, so maybe often you try to attach your biologics to any substrate, maybe which is used to deliver or to enhance the delivery of protein across the cell walls. Then the biological activity. So there the drug receptor interaction is very important. Then the PK profile, that after oral administration, how it is delivered across the cell wall, what is the bioavailability we are able to obtain. Okay. So I think the physical and chemical stability aspects we can briefly see. So when it is a physical degradation, then you see denaturation, precipitation, adsorption of protein onto some surface. In terms of chemical degradation, you have, depending on the molecular structure of that particular protein, it might undergo the deamidation, hydrolysis, oxidation, other components, and base. Once you have this idea whether my protein is sensitive to physical or chemical degradation, then it is possible to think around strategy or excipients how we are able to stabilize the protein molecule. So to summarize, the formulation is quite challenging from proteins, totally different from the small molecules because of its complex nature. The structure is quite delicate, so it's very, very sensitive to any degradation factor. And then it's also quite sensitive towards the external and environmental factors. So which are pathways taken by the proteins across the small intestine? So you have the possibility to, to deliver the proteins by using transcellular passive transport. So in this case, it's simply passing the cell line. But we'll see that it's not so easy. It's a paracellular, mainly it's happening between the two cells, and there are some tight junctions always. Maybe if you recall your pharmacology lectures from your B.Pharm syllabus, we know the tight junctions which are used normally to deliver the ions, but this can be also used to deliver the proteins across the cell. And then there's a possibility also to use a transcellular passive transport pathway. So here it was more active, here is more passive. So in case of active transport, you certainly need some energy, so it's more receptor coupled. In this case it's passive, but there we need some delivery device like nanoparticles, maybe liposome, etc. So what are the options or strategy we do have to deliver the biologics? So basically we have to protect the drug from the acidic enzymatic degradation in the stomach. The mucosal membrane, we have said, it's quite impermeable, so we have to make it more permeable. Basically also increasing the contact time of the drug with the absorptive epithelium is also quite helpful, because what happens, the epithelial clearance is quite high. So if we are able to increase that residence time, then it will help certainly to increase the delivery. And then the tight junction, just we saw that if we have any strategy to open the tight junctions by using permission enhancers, that is also going to help. This slide summarizes more or less the physiology of the GI tract. So esophagus, we have acidic pH, but the transit time is very short. When you go to stomach, low pH, gastric enzymes, then mucus layer is quite thick. So it's also quite challenging. Transit time is 1 hour. The maximum transit time we get mostly in the intestinal, the lower and upper intestine. The pH is more into the basic side, but you have some disadvantages. Surface area is quite low in this case. The bacteria rich environment which leads to degradation. In case of the small intestine, the variable gastric emptying, digestive enzymes are present, the mucus layer is quite thick, so this is to be considered. So which are the approaches we can use to enhance the gastrointestinal absorption? So there are some options available which are successfully used by researchers, and some of them are working, and as you will see, this has reached to preclinical or clinical stage. So we can think of co-administration of the protease inhibitors and absorption enhancers to inhibit the enzymatic degradation to improve the permeability. The physicochemical nature of the macromolecule can be changed to improve its permeability or stability towards the enzyme. Systems such as nanoparticles, microparticles, liposomes can be used to protect the molecules from enzymatic degradation. As we saw before about the the residence time. So to avoid any fast clearance, we can think of using the mucoadhesive drug release system to prolong the residence time. Site-specific delivery to colon is also possible, where the proteolytic activity is least, and product approach is another possibility. So let's start with the co-administration of the protease inhibitors. So basically, proteases are the enzymes present in the GI tract. They lead to the degradation of proteins, and that's the reason they are called proteases. Once the protein is degraded, it will get into converted to amino acids. And this is something we have to avoid. So these protease inhibitors, basically, they are just binding to the free form of enzyme. So protease enzymes, and it leads to the formation of the complex, which is inactive. So you can see this illustration here. So you are administering the enzyme inhibitor along with the biologics. You have enzymes present there. So once this enzyme inhibitor is binding to the enzyme is inactivated, and then your molecule is not degraded basically, and then you have the concentration gradient across the GI cell wall, and that's the reason that protein has the possibility to cross the cell wall, and you can expect some systematic bioavailability. So what should be our selection criteria for this enzyme inhibitors? So it should have very I'm sorry, it should have very good specificity towards the target enzyme. Compartmentalization is very important because we need this enzyme inhibitor at the subcellular level. It should have the capacity to reach to the target, and then how it impacts the absorption pattern of peptide, because imagine if your enzyme inhibitor is binding to the protein and that leads to inhibition of the bioavailability, so that's also not helping. Then another possibility is co-administration of permission enhancers. Maybe some of you know permission enhancers. We know quite often in the topical formulations where they try to increase the skin permeability. So more or less the same mechanism, but of course the choice of permission enhancers were different, and they act slightly with different mechanisms. So basically they try to disturb the epithelial membrane, which is quite tight, we saw before. So they try to disturb this membrane, so they try to make it more permeable or open. They also tend to open the tight junction, we saw before. They also try to reduce the viscosity of intestinal mucus. So what happens if your intestinal mucus is very viscous? So basically peptide is just trapped there and it has no possibility to cross the barrier, it's just clear. So when the viscosity is reduced, then it has a possibility to diffuse across the mucosa. It reaches to the cell wall and it can permeate, and it also leads to increase the membrane of fluidity, the membrane fluidity, sorry. So basically this is a mechanism. Again, you can see this illustration that biologics is administered. It has very limited absorption, but when it is conjugated to the permission enhancers, so due to these mechanisms, it impacts the permeability and there's increase in the overall bioavailability. So in this case, very important that the whatever permission enhancers we are using, it should be compatible with the proteins. It should not impact the nature of delivery vehicles, and then regional dissimilarities means it should really work as a permission enhancer across the length of GI tract. Any regional dissimilarity could lead to the variance of the bioavailability. As a case study, we can take the example of semaglutide, which is a GLP-1 analog, again anti-diabetic. The mechanism you can see here, it leads to lower the HbA1c, and which leads to facilitation of the weight loss. However, the commercial formulation is a subcutaneous. So as we discussed in the beginning of the talk, that for the sake of patient compliance, it's very important if the oral formulation is available. But when they were giving this drug by oral route, they saw the extensive degradation was happening by proteolytic enzymes in GI tract. There was poor absorption across the GI epithelium, and they tried to co-formulate of this drug with this permission enhancers, which is SNAC. So basically, it's a caprolate derivative, and it's important to note here, this is the first oral formulation of GLP analog which is approved in US. So very, let's say, fruitful outcome. And you can see the illustration how it works. So basically it's a pill containing the peptide attached to this permission enhancers. It dissolves into the stomach. Once it is dissolved, The permeation enhancer helps to permeate the drug across the stomach cell wall and it's reaching to the systematic circulation or systemic circulation, I say. This is the outcome of the clinical trial they did. It was done in Japan. So different clinical endpoints were evaluated, such as the weight loss, the decrease in the HbA concentration, and then specifically they have used the different comparators like insulin or metformin, the standard of care, I'll say in that case. So I'll not go into the details in the interest of time, but overall, what I can say that the clinical endpoint were reached quite well when they tried to use the oral drug, which was compared against the subcutaneous route. So change in body weight was one of the endpoint they used. Change in this biomarker HbA1c, and they try to compare, how this clinical endpoint is achieved when the oral semaglutide was given against the subcutaneous route. They also noticed that oral route had a favorable cardiovascular safety profile compared to the SC route. So it is not only helping in terms of the bioavailability, it is also helping in terms of the safety profile. Then modification of the physicochemical nature. Yeah? So in this case, we are trying to change the structure of the protein, so how it can help to improve the bioavailability. And basically, aim is here to improve the permeability and to inhibit the proteolytic degradation or to improve the proteolytic stability. So pegylation could be helped. So peg molecule, polyethylene glycol, can be linked to the proteins. It leads to improvement of solubility. It can extend the shelf life. Also, toxicity can be reduced. Immunogenicity can be reduced. And overall, it leads to increase the stability. The molecule is protected from proteolytic enzyme, and then there is no impact on the biological activity of therapeutic protein. So these are basically the advantages of pegylation. So you have the possibility of using branch chain pegs or the linear chain pegs, and then depending on the overall impact, you can decide. But majority of the companies are using the branch pegs. Then amino acid sequence can be also altered. So as an example, you can see here the L amino acid can be replaced by the D amino acid. So these are basically the stereochemistry we are talking, right? So those are stereoisomers. An example they have given the if they have investigated the analogs of LH-RH hormone, which was substituted at position 6 or 10 or both, and there they saw that the change in this the amino acid stereochemistry had a significant impact on the activity as well as the therapeutic action. So this is one aspect we can consider. Surface modification can also help. So basically, they are just trying to coat the protein with the bioadhesive polymer or surfactant, which can lead to improved bioavailability. So these particulate systems are also of importance, such as microemulsions or nanoparticles or liposomes. So basically, we are just trying to put the drug into these vehicles. That way it is protected from the degradation, and it also helps to pass across the cell walls. So that's a basic principle. The case study we can take of neural, which is cyclosporine. Cyclosporine, if you see the cyclic peptide, it has a molecular weight in the range of 1000. So its oral bioavailability is quite bad. So the commercial formulation from Novartis basically is from Sandoz, but Sandoz is part of Novartis now. So it's sort of Smith's formulation you can consider. So Smith's formulation put into the capsule, and that's how they try to increase the bioavailability of this drug. It's mainly used as immunosuppressant during organ transplants. So this is the PK profile. So this neural as well as Sandiman, these are both lipid-based formulation, but still you see some difference in bioavailability. Because in a nutshell, just to note down, the commercial formulations are of lipid-based systems. These days, in fact, there are a lot of reports also people are trying to use a solid dispersion formulation also to increase the bioavailability. But the most successful formulation so far was the lipid-based formulation. Then the mucoadhesive systems, we discussed before that the clearance is quite high. The molecule is not in contact with the epithelial surface, and that's one of the reason then the bioavailability was not so good. So they tried to use the mucoadhesive polymers such as chitosan. So you see here, the molecule is encapsulated with chitosan, and then due to the mucoadhesive nature of the chitosan, it binds to the mucosal surface. The contact time is quite high, and that leads to the increase in bioavailability. This is the principle normally. So basically we are just trying to increase the concentration gradient across the cell wall due to this longer residence time. And this, it basically forms the hydrogens, right? Because this mucoadhesive polymers, they have the ability to take the water molecule in, it swells and the hydrogel is formed, and they are able to react to the different changes in GI tract, such as pH change. This is very trigger. You must have seen a lot of carbopols or ketone sands, maybe some eudragids. They are quite pH sensitive, so you can really tailor your delivery system based on the pH you are going to target, based on the molecule you have. So depending on the molecular trigger in vivo, you have the possibility to trigger the release of the drug as a function of pH, light, electrical charge, or any. You can also take into account any biochemical changes in vivo. And then site-specific delivery to the colon. What are the advantages? So enzymatic activity is quite low in the colon. The response is quite good to the permission enhancers. Transit time is also quite long. We saw before, the residence time is quite good in colon, and the environment is quite alkaline, not acidic. In case of pH, the stomach pH, we have seen it's 1 to 2. In case of the colon, it's quite on the basic side, 67 or maybe more than 7 in some cases, and that is quite favorable to the proteins. And this pH we can use as an advantage to deliver the proteins. So basic materials we have like eudragids, this can be used as because eudragids are known to work as an alkaline pH. So basically they are not dissolved in the acidic pH when the formulation is passing through the stomach, but at internal pH they can get dissolved and molecule can be delivered. But you do have some limitations there. The surface area available in colon is quite less. So maybe you have seen somewhere that the disease condition like geriatric patients, their colonial flora or colonic microflora is quite damaged, and due to that then the absorption is badly impacted, and then this could be one concern, and this should be avoided. Sorry. Then product approach, so it's more or less same as we saw in small molecules. So basically we try to create a product by attaching a moiety on the surface of the biologics, so and it is administered. When it is administered at the site of, let's say, the cell passage, the molecule is cleaved again into that moiety in drug, and that leads to the increase in the bioavailability. So we are preparing products for two reasons: to avoid its degradation against protease, and also to increase its permeability. So whatever principles you have in your mind for the small molecule products, this can be also used for the biologics. But although, let's say, the making products of the proteins or antibodies is not so easy as it could be for peptides. So this is also one thing we have to keep in mind. Then let's talk about cell-penetrating peptides. So in this case, basically biologics, the target biologic molecule like peptide protein can be just coupled to the cell-penetrating peptide. So cell-penetrating peptide is basically peptide molecule. So it has its own ability to across the cell layers. So when it is coupled to the biologics, it leads to increase in the formation of the overall complex, and once the complex is entering inside the cell, again it's cleaved into the CPP, self-penetrating peptide, and the molecule, and that's how it is delivered to the systemic circulation. So so far we studied about the different options we do have to deliver the proteins. Now let's talk about the technologies that people have explored to deliver the oral insulin. So because I think insulin is the molecule which has been most widely studied by the pharma companies to be delivered by the oral route. You'll see a lot of literature, of course, and they are quite successful as well. So using targeted approach, you will see the option of using the receptors. Then they also tried to use CPP such as penetratin, octarginine. Then this smart oral robotic system, Soma, will see in detail later on. Lumi also tried to cover into this presentation, we'll see. So basically the medical devices we can put it together, and you do have the option of using the nanoparticles or hydrogens. So if you have any biological molecule in your mind, you want to deliver by oral route, you can take into account these options. It is possible to put into one of these technologies. Of course, it will be never one to one, but depending on your structure of your molecule, you can really think which option can fit best. So what are the platforms? They are already into the clinic. So again, you'll see most the insulin remains the most successful molecule. I'll not go into the details for every technique, but you can at least see the technology they have used so far. These are all, of course, patented by these innovator companies, and they have successfully evaluated if insulin can be delivered by the oral route. For example, here they have reported for octreotide, which is a peptide molecule. Then salmon calciton is also, let's say, peptide. And here we have heparin, the protein molecule. So there are options to deliver this molecule. So here GPET, it has been used for insulin as well as the GLP-1 agonist. So GLP-1 agonists are peptides normally. They are much small in molecular weight compared to the insulin. And about oral peptide technologies at the different stages of delivery. So all the way you see it starts with the phase 1. So you do have the calcium phosphate nanoparticles, then use of the antibodies. You have penetration answers reaching to the phase 2. Phase 3, you have penetration answers, product approach, the option of the cyclic structure, entry coating, and then you already have some technology into the market. For example, insulin, you have seen the neurals, and you mean other cases they have delivered the small lipopeptide, cyclic structure, entry coating, etc. And these are already into the market. And again, I tried to segregate what is for local delivery, what is for systemic delivery. So you have the names, you have the technologies. If you are really interested, you can go into the details, and then we try to get sort of concepts, what concept they have used, to what type of molecules they try to deliver, what is a particular dose. So depending on that information, it's easy to really figure out then whether the same technology approach can work in our case. And here it's a summary of the commercially available oral peptides, just to highlight. These are all given by the oral route. So whatever molecules you see on this slide, for all these peptides, there are formulations onto the market which are given by the oral route. So which is, I think, quite successful approach considering the complexity of delivery of these molecules. And you can also see that there's formulation from Biocon for this colistin sulfate. And then on this slide, I tried to put together the information for the oral peptide formulation in phase 3. So these are not yet onto the market, but at least the phase 3 trials are going on. So whatever concept they are using, at least there's a proof of concept that it works to the clinic, and most likely it will be successful. This 5-SNAC we already saw before for semaglutide chemical modification against from Biocon. So they tried to use it for insulin. And the approaches are quite important. Sodium caprolate, again, I think it's the penetration answers. Local pH modulator, we saw the example of idragits, etc. So by the virtue of making the local pH change, you can try to reduce the protein degradation. So let's talk a little bit about the devices, the option we have to deliver the drugs, by using the sort of medical devices. Example we can take of SOMA, self-orienting millimeter scale applicator. So this was, as I said in the beginning, this was discovered by in Bob Langer's lab in MIT. They are in talk with Novo Nordisk, and this has already reached to the preclinical phase, and soon we should have the clinical data. So it's quite an interesting concept. They try to take into account the shape of this African leopard turtle. You can see the shape. So when there's any stimulus, it tries to go into this oval shape, right? And that's how they try to develop their device by using simulation studies, and then in practice how it appears. So basically you have needle, you have the protein reservoir, in the end you have some water soluble molecule, maybe sugar. So how it works basically, so when it is administered by oral route, so it tends to find a stable conformation. So in this case it's unstable because it is not really in its most stable form. But when it is facing this intestinal cell wall, which is basically the most stable conformation it can find. And once it finds the most stable conformation, the sugar molecule is getting dissolved, which leads to the stimulus to this spring, which or the needle, I'll say, and which is followed by the delivery of the biomolecules, which is present into this reservoir, and that's how the actual delivery of the biomolecule is happening. So on this slide, we see the force of penetration of this needle inside. So how many millimeters it is piercing across the epithelial cell wall. You can also see here, basically this is a histological diagram, and you can see here how inward it looks like. So this is the needle which is piercing. Once the sugar molecule is dissolving, which leads to the trigger of the needle activation, and that's how the molecule is being delivered. And on your right, you see the delivery, data on the delivery. So there they try to compare the data using the subcutaneous injections against this soma device. And basically what you can interpret, the delivery of the drug by using the soma device was as good as the subcutaneous injection, and then you have the possibility to replace the subcutaneous injection by this oral soma device. Then the next option we do have the luminal unfolding macroneedle injector. So these macroneedles maybe you have also seen in case of transdermal drug delivery system, where they try to put the macroneedles on the surface of the skin, which leads to the formation of micropores. And once the micropores are formed, the drug can be delivered. So it's the same concept here. So basically, they try to incorporate the macroneedles into the pill, and basically in the pill they are all, if you see, they are more or less aligned, but when it is reaching in contact with the cell, the intestinal cell lining, then basically it's opening or distorted, and that leads to the formation of, let's say, the the pattern which can really penetrate across the cell walls, or which is piercing, creating macropores across this epithelial cell lining, and that leads to the delivery of the drug across this cell lining, because the pores are being formed because of the microneedles. That's a basic principle. So here is again, it's a illustration here how it works. So the enteric coating is dissolved. So basically it's happening in the intestine. Once it is dissolved, this the spring actuator is mainly dissolved here as a follow-up step of enteric coating dissolving. Once the spring actuator is dissolved, the device is propelled from the capsule, so it's coming out basically, and when it is coming out, it's opening, and then it is forming the micropores into the epithelial cell lining. Drug is coated onto this microneedles, and due to that, the drug is getting permeated across the cells. So that's the overall principle. Here you can see the image how the microneedles look like when they are into the capsule, where the spring actuator is not actually activated, but once it is actuated, the deployment of this microneedles is happening. And on your right, you see some PK data, how the PK profiles are obtained using this Lumin device. You also have the impact of human insulin delivered. So there's a head-to-head comparison they have given of Lumi compared to the patch, microneedle patch, compared to the subcutaneous injection of the molecule, and more or less the data is not quite significant. So we can say that it leads to the statistically non-significant delivery of the molecule by using this technology compared to the subcutaneous route. Then this is brief summary that what it costs basically when you try to develop the biosimilars and biobetters. So the investment is quite huge. We already discussed in the beginning. So for innovators, more than 1 billion, but when you are talking about biosimilars, more or less 100 to 300 million dollars. So of course that's the reason more and more Indian companies are after biosimilars rather than the innovators. In case of biosimilars, it goes from $300 million up to 1 billion again. In case of biosimilar, more work, biosuperior, more work is needed, because it's not just a similar biomolecules, but we are trying to work on the protein engineering which may lead to the molecule with the better activity, better safety profile. And going into the novel drug, which is totally different from the innovator or this stuff, again, significantly investment is needed, basically more than 1.5 billion US dollars. And that really depends which target we are going to approach. The target needs to be validated to make sure the best possible clinical outcome is obtained. And why we need that? Basically, we want to improve the potency of the drug, half-life could be improved. We want to have the better safety profile, less immunogenicity, and broader efficacy. I can also try to take one example, oral vaccine delivery. We discussed in the beginning about corona vaccine, that it's parenteral, it should be sterile, and that is having impact on the supply chain. Why can't we make oral? But that's not so simple. Basically, we do have some oral vaccine to the market. You can take example of the polio vaccine. I think most of us have already taken. It's so easy. They just put 2 drops in the mouth of a child and it goes quite smoothly, right? But unfortunately, it's not still the case for the current vaccine program we have. Maybe in future, why not? But again, I mean, it really depends on the molecule which target it is going to approach, how stable it is, whether the immunogenic response is obtained, etc. But I mean, this is one option. We can always think of any vaccine molecule, whether it can be delivered by oral route. And you do have the vaccines already into the market, polio, rotavirus, typhoid, or cholera. And this is basically the schematic illustrations when the vaccine is delivered by oral route, where basically it's passing by, how it is going to elicit the immune response. And basically it's trying to target the lymphatic system, into the mesenteric lymph nodes. That's where basically it's trying to create the immune response. So what are the options we do have for the oral delivery vaccines? So we can think of membrane vesicles such as liposomes, nanoparticles, or even the emulsions. So the vesicular system we can think of using for the oral delivery of the vaccines. And this is a case study of the, let's say, the example from the market for the typhoid vaccine. So basically these are enteric-coated capsules containing this protein, viable salmonella typhi. That you have units, how many colony forming units are there. The excipients listed there, the amino acids, ascorbic acids, the standard capsule excipients, I will say. And here, basically, it's a mechanism that when it is given by oral route, where it is going to dissolve, where it is going to react, to generate the immune response. So, what are the challenges? We do have the clinical translation of the oral deliver the biologics. So when we go from bench to bedside, so when I say bench to bedside, we can do a lot of expense in our lab, but at the end of the day, you want that formulation to the clinic or to the market. So we need to have the control design performance. It should be robust. Maybe that it works quite good in the lab, but if we are not able to scale up, then it's not going to work. The safety should be acceptable. Maybe in early research, we have a lot of liberty of using any excipient, but when we go to clinic, FDA must accept that, right? Scale up, I already discussed. Then cost benefit ratio. I mean, if it is too expensive, then nobody's going to buy it. So this is also one thing we have to expect. And then we have to focus on the toxicological regulatory point of view. So whatever we use in terms of the excipients, technology, packaging, it must be acceptable by regulatory bodies, especially US FDA. There should not be any toxicological concerns. Now you see the example, Pfizer wants to bring their vaccine to India, but there are a lot of negotiations are going on concerning the toxicological profile. Indian authorities are asking, please show us the safety data in Indian population, which maybe Pfizer do not have at the moment, or they, I don't know. I mean, there are some negotiations still ongoing. So that's what I would like to highlight, that regulatory bodies must be satisfied regarding the safety or toxicity of your technology or molecule, and without that, it's not possible to launch the product. So no matter how good is your product, but if the regulatory bodies are not allowing, you are simply not able to launch it. So regarding the dosage forms, we need a solid dosage forms, maybe using freeze drying, although it's very expensive. So for freeze drying, you need to add some cryoprotectants, and we need to maintain the bioactivity of the macromolecule into the final formation. So basically, this is to be taken into account when you want to develop the oral system for the biologics. So with this, then I would like to conclude just in time. So as I said in the beginning, 50 minutes I'll take for the talk and 10 minutes I'll keep for question and answer. So To conclude, we have the few clinically tested technology today for the oral delivery of biologics, but there's long-term research activity still needed to scale it up so that we have the possibility to overcome the barriers we have seen in the beginning of the presentation, in terms of the proteolytic activity, unfavorable pH, the viscosity of mucus lining, etc. for the successful delivery. There are many chemical enhancers used. We saw the examples of caprolates, which can help to decrease to deliver the biologics across the cell. And in vitro screening is possible there. You can do some in vitro studies to show which penetration enhancer is working. Nanomedicine has shown a lot of hope in this area. Basically, if you see the Pfizer and Moderna vaccines, which are being used in Western world, they are still not in India. But what are corona vaccines they are using, Pfizer, Moderna, they are based on the nanoparticles. So they are using basically the solid lipid nanoparticles. So the lipid is being used there, which is to deliver this mRNA-based vaccine. So I think this is going to get some importance. Already it is getting importance, but now it will be more and more. So in these areas, basically the oral biologics delivery, more and more research will be certainly needed, because the parental delivery certainly put in a lot of it puts some burden onto the supply chain, the cost, and of course affordability into the developed world, because not all the countries can afford such a cost and the delay. So oral route should get more important in this case. And then other aspects should be coming into the picture: electronics, robotics, or material. We saw the simulation there, so that's what I tried to put these aspects into here. So with all this, then we have the possibility to increase the delivery of biologics across the oral route. So with this, I would like to conclude my talk, and I'm more than happy if you have any questions.
Yes, sir. There are a few questions, so we'll take one by one. So here is the first question, sir. That please tell regarding nanostructured lipid carrier.
Uh, can you show me the question? Maybe I can see on the screen, or you can repeat. Okay, I
So
see.
please,
So it's
yeah.
fine. Okay. So I mean,
Yeah.
nanostructured lipid is basically we are. Can you put on mute, because then I see a lot of echo. So basically, in case of the nanostructured lipid carrier, we are trying to use a solid lipid. We also call it NLCs. So basically, in that case, the lipid is I mean, it's typical SLN technology, I will say. In that case, your drug is in contact with the lipids, and of course, you will also have a surfactant there. Surfactant is really helping to get disperse your lipid molecule into the aqueous component, and that's how you will aim to deliver the molecule, whether it's small molecule, large molecule, doesn't matter. The concept should be still same.
Okay. Uh, Now the next question is this one.
There are some successful examples, small pills of peptide therapy. I mean, maybe I'm not getting your question, because there are a lot of successful small molecules, peptide molecules, right? So I don't know whether you're asking in terms of the technologies. So technology we discussed for semaglutide, they have used the caprolate as a penetration answers. For insulin, we have seen how this Soma and Lumi technology they have used. For cyclosporine is not a small molecule, in fact, but you can consider it maybe small molecule sort of. So there you have seen the lipid-based systems are working quite well. Charges on polymer can affect the mucoadhesion. If yes, then what will the solution? Okay, I mean, then first thing you can really try to see whether it's possible to use the non-charged or uncharged mucoadhesive polymer. Or in that case, you can also think of using the oppositely charged ions. If your polymer is negatively charged, what happens if you try to use the counter ion to balance that overall charge? That's one possibility you can think of. Well, they have not revealed much regarding Soma. If you are interested, you can go to MIT site. But it's it's not basically much regarding formulation. It's more the technology. So as I told you before, they have this turtle type of device which is really composed of your molecule which is sitting in the reservoir, and at the front they have the sugar molecule. So sugar could be anything, so you can even imagine a sucrose. Basically you need something which should be quickly dissolved, which is creating in osmotic pressure. Due to that osmotic pressure, that needle is activated. And once needle is activated, it is just piercing across the epithelial cell line, and that's how the delivery of the peptide or protein is happening.
Okay, so Dr. Suhas want more details about
The
this
drug
process.
sorry,
Yeah, actually, Dr. Suhas want more details about this process.
which process?
That is this the drug device, the release of drug to drug penetration.
Maybe I can go to the slide again if you want. Do you see my screen?
No, no, sir, not yet.
Uh, now,
Yes, sir.
okay. So I mean, as I said, you will, it's a patent technology. Not all the information is available. Something quite similar to this turtle polymer, so which is as long as biocompatible, it should not have any adverse events. And once this Hello device is made, you have the possibility to create a protein reservoir here. At the front, you would like to have your any water soluble molecule like sugar, huh? And you also have the needle there, which should be, and this is more, let's say, the mechanical component, which is not really any excipient. I will say it's more your device component. So basically what's happening when it is finding its most desired simulation here, when it comes with the fluids or water molecules here, the sugar molecule is getting dissolved. So basically the surface layer is getting dissolved. Once it is dissolved, it's actually activating the needle component there. Once the needle component is activated, it is piercing across the cell wall. Once it is piercing the cell wall, the pore is being formed, and that's how it leads to the delivery of the biomolecule which is sitting into the reservoir. And once the molecule is delivered, then the device is just excreted as any foreign body from the intestinal tract. I hope this answers your question. Can peptides are more active therapeutically than small? Yeah, naturally yes, because the mechanism action is quite different. So that's the reason you will see the dose of the peptides protein is very small. So when you have, let's say, a small molecule 500 milligram or 1 gram dose, in case of peptides they work even at microgram quantities. Often for many peptides your dose is in the range of 50 to 100 microgram. But your point of view, which is good crab? I mean, there are a lot of very successful cryoprotein like sorbitol or trialose. I mean, a number of cryoproteins. You go to FDIG database, you will find it. It also depends on your molecule specifically, how it is acting in contact with your molecule, whether you are able to get the very good lyocake. But there are a lot of possibilities.
Okay, sir. So we'll end up with the question and answer session. So now we will conclude the session. So I request Shweta madam to please conclude the session. Before concluding the session, herewith I'm going to inform the participants that the attendance link is shared and it will be active till 3:30.
Okay, then thank
Shweta
you very
madam.
much.
Thank you, sir. Uh, Thank you, sir. Thank you, Dr.
Thank you.
Yogeshwar Bachcha, sir, for this dynamic session on oral delivery of biologics. Sir, very well explained about the oral drug delivery strategies Then sir also talk on about the technology platform for the clinical applications, oral peptide technology at the different stages of the development. Sir also different devices available to drug delivery, and sir also focused on the challenges of clinical translation of the oral biologics. So again, I would like to thank Dr. Yogeshwar Bachcha, sir, to visiting and enlightening us with your knowledge. Um, Now it gives me immense pleasure to convey the vote of thanks for the 2 days session. Here I thank to all our guest speakers of the morning session and the afternoon session. All sessions are very informative and helpful to our all faculties and the participants. I would like to thank the management of the Konkan Ganpit Rahul Darkar College of Pharmacy and Research Institute for providing us a platform for conducting this online faculty development program. I thank to Dr. Mohan Kale, Principal Sir of the Konkan Ganpit Rahul Darkar College of Pharmacy and Research Institute, providing guidance. We also thankful to AICTE for sponsoring this online faculty development program. I also thank to all organizing committee, and I special thank to Mr. Ajay Kharche sir for technical support. I sincerely thank all the respective faculties from different colleges for patiently attending the session on day third of online faculty development program. Thank you all. With this, I conclude this session. We shall meet tomorrow with the session of the day fourth. Morning session begin at 10 a.m. So I request all to join 10 minutes before the session. Thank you all.